Long, long ago, in 1973, in a land far, far away, a mad and evil professor was carrying out his greatest experiment yet. An experiment that would change the course of history and allow him power no man had ever had before. The power to combine species. And what is his experiment, you ask? Well, he will be injecting the unfortunate bacteria before him with lizard DNA. He marks this day as the first official step in genetic engineering. But does all go well for this professor? Does everything go according to his immaculately detailed plan? Oh, it goes well. A little too well than the professor had cared to imagine. You see, the unfortunate bacteria, once it was injected with the DNA, began to take on the lizard's regenerative properties. It began to multiply furiously in great numbers, forming a bacterial monster on the table. The professor, watching his creation, initially thought that things were going well, but soon realized that something was very, very wrong, and that his creation must be stopped. But even as he had the thought, the bacteria reached for him across the table. In a panic, he grabbed the nearest object beside him and thrust it at it. However, this was a mistake that would cost him his life. But he had done nothing but provoke it. The bacterial monster formed a giant mouth, fangs dripping poison, and launched itself across the table at the poor professor. In a single leap, it was on him, covering his body and silencing his screams. And so began the reign of the superbacteria. That's not quite right. What really happened was that two very smart men discovered the structure of DNA. Then they found enzymes that could extract specific genes from strands of DNA, known as chromosomes. The genes were then inserted into another organism by another enzyme. Genetic engineering was born. This is still a very controversial topic. Here are some pros and cons.
Those in favour claim that it's very quick and very precise. Only genes that are required are transferred from one organism to another, and these genes act in a precise way. So, for example, the gene for the manufacture of human insulin is transferred from a human cell to a bacterial cell. The bacterium will make human insulin and not its own version of insulin. Another enormous advantage illustrated by this example is that, unlike in selective breeding, there are no species barriers to this process. Genes from humans can be transferred to bacteria, and genes from fish can be transferred to strawberries. Those who argue against genetic engineering do so for a variety of positions. Religious people claim that we should not be tinkering with God's creations. Others argue that genetically engineered organisms may pass their altered genes to wild species and bring about unpredictable changes. Also, can we ever be certain that genetically engineered plants and animals are safe to eat?